39 amino acids and your product is a drug. 41 and it's a biologic. Same chemistry, same instruments, same team, two completely different regulatory worlds. Today we look at the line that separates them and why counting is the highest value thing you can do early in the peptide program. Hello, I'm Raghuveer Singh Parmar, Director Scientific Affairs at Resolve Mass Laboratories, and we work with pharma and biotech teams to build the analytical packages that peptide and protein submissions stand on. Today we are going to walk through the 40 amino acid line, what it says, how to count, and what changes on each side of it. If you have found anything valuable in this channel, please like and subscribe this channel. This gesture of appreciation really means a lot to us. For a long time, peptide and protein were loose words. Textbooks drew the line somewhere near 50 residues and nobody had to be exact because the labels carried no legal weight. That changed with the Biologics Prize Competition and Innovation Act in 2010, which created a follow-on pathway for biologics and a 10-year window to move certain products across. FDA proposed a hard definition in 2018 and finalized it in February 2020. On the 23rd of March 2020, the line went live and the word became a number. Here is the exact wording. A protein is any alpha amino acid polymer with a specific defined sequence that is greater than 40 amino acids in size. Read it slowly because every clause does work. Alpha amino acid polymer sets the chemistry. Specific, the defined sequence rules out heterogeneous mixtures where you cannot write one sequence down. And greater than 40 is the count. 40 or fewer, your product is a drug. 41 or more, it is a biological product. There is no gray zone in this text. So how do you count? For 4 rules. First, count amino acid residues, not molecular weight. A heavily modified 30-mer is still a 30-mer. Second, when a molecule has more than one chain held together the way it happens in nature, add the chains together. Insulin's A chain is 21 residues and B chain is 30, that is 51. So insulin is a protein. Third, lipid side chain, PG and sugars, and add mass, not residues. Fourth, sequence has to be defined. So now the consequence, at 40 or fewer, you are in the drug world. An NDA or 505(b)(2) and a follow-on can come in as an ANDA that shows the same active ingredient. Above 40, you are in the biologics world. A BLA and a follow-on has to come in as a 351K biosimilar showing no clinically meaningful differences. Different applications, different review division, different exclusivity. Same molecule, one residue apart, and keep in mind this is a US line. In the EU, whether something is a biological medicinal product turns on the source material and how it is made, not on a fixed size cutoff. A global program can carry 2 different answers at the same line. The classification does not stop at the cover page. Your CMC dossier changes shape. A peptide file is built around synthesis, purity, and impurity control, while a protein file is built around the cell line, the process, and comparability. your sameness bar changes, and your life cycle changes, exclusivity, post-approval change rules, and interchangeability all run on different clocks. This is the step where the work is easiest to underscore because the count feels like a labeling detail right up until it sets your budget. If your molecule is sitting anywhere near this line, drop its residue count in the comments. Those replies say what we cover next. Now, 3 real molecules make it concrete. Insulin, 51 residues. So on the 23rd of March 2020, it was deemed licensed and its NDA became the BLA overnight, along with the somatropin and around 90 other products. Tirzepatide, 39 residues, 1 under the line, so it stayed a drug and was approved through an NDA. Teriparatide, 34 residues with a reference product made by recombinant DNA, and because the molecule sits under 40, synthetic generics were cleared as ANDAs in November 2023. At Resolve Mass, we have supported sponsors through exactly this kind of questions where the residues count reshape the whole submission plan. If you land on peptide side and want the generic route, FDA's synthetic peptide guidance sets the bar, and it is specific. Sameness means comparing primary sequence, higher order structure, aggregation, and biological activity against the reference product. Impurities carry hard numbers. Any shared impurity has to sit at or below the reference level. A new impurity above 0.5% is not acceptable, and anything between 0.10 and 0.5% has to be identified, characterized, and justified. Add an immunogenicity risk assessment covering both innate and adaptive signals, and that is the package. So, what should you walk away with this discussion? The 40 amino acid line is a legal boundary, not a biochemistry convention. Count residues across every chain that occurs together in nature and ignore anything that is not an amino acid. The count sets your pathway, your sameness bar, and your whole analytical package. So do the count in discovery, not at the pre-submission meeting. If you're developing a peptide or protein and you want a CRO partner for this kind of projects, please contact us, Resolve Mass Laboratories. Resolve Mass Laboratories supports small and mid-sized pharma teams from the early characterization and impurity profiling all the way through submission. Thank you so much for watching this video till the end. Before you go, I want you to do few things. Number 1, subscribe this channel right now. The more you subscribe, the more valuable pharma insights, expert tips, and industry updates we can bring to you. Please comment your feedback. It will help us to create and improve our videos and content. And also share this video to at least one person who you think would find it useful. Thank you so much for watching this video again, and see you in the next one. Until then, goodbye for now.